Hi, how are you doing? Today we're going to be talking about our first major lesson, introducing biology. First thing I want to hit on is taking notes. So every time I give a lecture, I'm always going to have something like a big question. So this is an idea. You want to take a couple minutes to think about what the importance is, and by the end of this lecture, you want to be able to say the answer to it. So today's big question is, how do you take notes? So take a moment, think about that. How have you taken notes in the past? And the second question is, why is taking notes important? So, just a quick overview I wanted to talk about. There are two major formats for note-taking. Outline, so this would be kind of your bullet points, and Cornell notes, which is the kind that we're going to emphasize and hope that you'll take in this class. So what's an example of outline? So outline alternates letters, numbers, to show points. So from student data, we found out in some of my classes that a majority of students like football, and the least likely sport is basketball. So notice that these ones are details, and over here we have big ideas. So you go from big to small in outline. So how to summarize, how do you do an outline? You use letters or big ideas, and you continue on with the numbering. An important part of this when you're going from an outline is to annotate. So what does annotating mean? It means highlighting and emphasizing the important part. So for example, you could use an exclamation point for something that you find is exciting or interesting. You could use a smiley face for something that you think is helpful. You could make a question mark for something you're concerned about or you actually have a question on. This can help you later when you're studying. So what do Cornell notes look like in contrast? They look like this. You split up a third, 2/3 of your notes. You also have a summary. That's about 3 to 4 sentences, and we have an essential question. So this is what makes it different than maybe some of the other notes you take. So what are the parts? So again, we have the big idea at the top. The left 1/3 is used for vocab, study questions, and big ideas, and the right 2/3 is for your regular notes that are usually done in outline format or bullets. At the end, you always want to make sure you write a 3 to 4 sentence summary and reply back to the essential question. So if you remember, the essential question was how to take notes and why is it important to take notes. So why is it important to take notes? It can help with processing and recall if you're a kinesthetic or verbal learner. For example, I'm kinesthetic in that I always need to write down what I see or hear, otherwise I will forget it. It also helps you focus on what's most important. By summarizing it, you figure out what's the most important part that needs to be memorized. It also is helpful for using a study guide. Remember, I talked about annotations. Annotations are helpful for that reason. It also creates a record of your progress, and it references it for further use. Say tomorrow you don't remember what we did in class, you can reference back to your notes. It's also an opportunity to become engaged, especially in this flipped classroom learning, by writing down questions and, of course, always making connections. So now for the meat of the topic, today I'm going to talk to you about the introduction to biology. So first off, take a moment and think, what is biology? And secondly, what does it mean to be alive? This will be especially important when we get to our lab, the life lab. So first off, what is biology? Biology is the study of life, bio being the root meaning life, and ology meaning the study of. So what does that include? It's the study of plants, animals, genetics, development, anything that's living, including things like cells that are part of biology. So what are some more examples of cells? Here's some cells that I stained in my lab, and here's one that you might find in a typical textbook. Cells are the base unit of all living things. So, just to check for understanding, what do you think biology is? Take a moment, think about it. You thought about it? Biology is, again, the study of bee life. So think, what makes something living versus non-living? How do you know? How do you know? For these reasons. So here's 3 of the reasons. If it's made of cells. If it's made of cells, it's considered alive. Even something that's made of only one cell is considered alive. These are some key words, unicellular or multicellular, that you're going to need to know for the life lab. In order for something to be alive, it has to reproduce. It has to be able to have children. And it also has to have metabolism, meaning it makes and uses the energy. So you might think of things like plants and animals. Plants make their own energy, and animals use the energy from the plant. What are the other reasons? Homeostasis, heredity, responsiveness, growth and development. Homeostasis means it self-regulates. The perfect example is in humans. What happens when you get too cold? You shiver. And when you shiver, that helps raise your body temperature. Heredity means you have DNA and RNA that can be passed on. DNA is this crucial molecule. It's a crucial molecule that determines what traits you're going to have when you grow up. 6 is responsiveness. If I poke you, what do? You respond. You say, hey, get that out. Don't do that. So you want to respond, react, and adjust to your environment. If it's too cold, maybe instead of sweating or trying to warm up, you put on a sweater. That's another way that you can adjust to your environment. Lastly, you have to grow and develop. If you're truly alive, you're going to develop and get either bigger, more complex, or change. So remember, you're not the same person you were in kindergarten. Everyone's going to grow and develop. And they do that by either working hard, changing their cell developmental plan, or learning and changing their neural plan. So growing and developing is the last portion of what you would be to be alive. So let's review. Which trait is not a characteristic found in all living things? Is it A, they move from place to place, B, they all have cells, C, they all need energy, or D, they adapt to their environment? So let's think about this as a multiple choice testing thing. They all need energy. Are you alive? You need energy, so it can't be C. They adapt. So humans are really famous for being adaptable, so it's not D. They all have cells. Remember, cells is what all living things are made of. So it can't be cells. How about they move from place to place? It must be that one because that's left. But can we think of a counter example? What is a living thing that does not move from place to place? One example is sea anemones and plants. Sea anemones are sessile, meaning they're anchored to the ground and they stay put. The same is true of plants. That's due to the structure of the roots. Here's some other vocabulary you'd want to know for the life lab. Autotroph is something that makes its own food. Remember, metabolism is a characteristic of a living thing. Auto, think of an automobile, means it does something on its own. Auto is the root for own. What's the opposite of an autotroph? It's a heterotroph. So heterotrophs are different in that they eat other organisms. Emphasis here on other. If you're a heterosexual, you like the other sex. So heterotrophs are organisms that eat other things. For example, humans and panda bears. 2 more vocabulary words to hit on. The first one is vertebrate. So if you're a vertebrate, you have a spinal cord and a backbone. So these tend to be your organisms that have a more complex nervous system because of the spinal cord. This happens in a step of development called neurulation, where you develop your nervous system. In contrast, the opposite of a vertebrate is an invertebrate. So this is where you don't have a backbone. So think of something that doesn't have that bilateral symmetry, bi meaning 2. You're not symmetrical on both sides. An example would be a sponge. Sponges is a living thing, or earthworms, which have radial symmetry, meaning that they're the same direction, when you cut them in half. So here's some quick summary questions. Can you list all the 7 characteristics of living things? Think about how do living things interact with each other? How do non-living factors impact living organisms? And why can movement not be used as a characteristic? So we'll take a moment to think about those and then we'll explain it. So 7 characteristics, you can look back in your notes. How do living things interact with each other? Think back to the words autotroph and heterotroph, and how those words relate organisms. How do non-living factors impact living organisms? Try to think of things like the weather and the environment. Lastly, movement cannot be used as a characteristic because there are exceptions to the rule, such as plants. There ends the end of characteristic living things note. So before we go into the lab, it's important to give you a couple reminders about the scientific process. So a lot of people see the scientific process as one linear stage, but I wanted to show you that science is not linear. It's not always linear. It's actually quite cyclical. But if you wanted to put the steps in order, you tend to think of them this way. An observation is where you see, hear, feel, or taste things that you have questions about. So for example, you might see something like a platypus, and you wonder, is that thing alive? Then you make a hypothesis. A hypothesis is an educated guess based on observations before the experiment. So you may wonder, oh, is that platypus alive? I think it is alive. If it is alive, then... So most hypotheses are in an if-then format. And you can make a prediction about that. So when you make an educated guess, what is it? Is it a principle, a theory, a hypothesis, or a conclusion? Take a guess. It is C, a hypothesis. Hypothesis is an educated guess in that if-then format. If it is alive, then it will do this. This is not a theory. Theories are special in that a theory, this is pretty important, I'm taking the time to write it down, is a explanation for observed phenomena that can't be tested. So some people think, oh, the theory of gravity. We can't test our hypothesis. We can't say if gravity exists, then there's no statement that works for that. A theory is the best explanation that science can come up with for all the things we say that's widely accepted based on evidence. Science is all about the evidence. And we'll get into that as the year continues. Continuing on, when you're looking at your experiment, there's different variables. So for example, there's a control group. So your controls tend to be, there's no real test on it. If you want to see if something happens or doesn't happen, that'd be the control group. You're double checking that your procedure is good. Experimental group is the one that you're affecting. So in that, you're going to do two things. The independent variable, which is the factor you're actually testing, and that's the thing you're giving them a drug or you're not. And the dependent is the one that you're measuring. So if you're going to put this on your graph, on your x-axis, so that runs horizontally, this would be your independent. So this could be things like time, drugs, amount of light. Your dependent variable is your y-axis. This is the thing you're trying to create a relationship to. There are different types of data. These are just two vocabularies. Qualitative data, data with numbers, only words and descriptions. So a way that I try to remember is qualitative is for letters and quantitative is for numbers. So for example, if I were to tell you a story about what a platypus looked like, it's brown and it's furry and it's a duckbill, that's qualitative. If I told you I found 22 platypi, that would be a quantitative data or a number. So taking a look, a computer model of cellular mitosis can simulate the aspects of division well. However, microscopic observations can improve understanding because actual observations, so this question is asking you to compare something in a computer versus something in real life. What do you think? Take a moment. And you should have come up with A. They may reveal greater unknown complexities. Real things are different than simulations. A simulation can help you study things that are difficult to observe. So, for example, if people want to study evolution in dinosaurs, they might have to make a simulation because they don't have time to. In this class, we'll be doing a lot of simulations because we can't actually go outside and look at things like volcanoes. We can't actually go outside and make mice mate with each other. So we have to simulate that in our labs. So far, as we've gone through the process, we've had observations, we've had a problem, we've had a hypothesis, we've had the experiment and the data. What do you do after that? After that, you're going to analyze it. So that means you're going to take information from it and you're going to do something with it. So you can create graphs, tables, you can evaluate it, say if it's true or not, if it has biases, and after that you're going to make a conclusion. So the analysis of the data and conclusion are the most important part of the experiment. All this stuff is for naught if you don't have analysis and conclusion. Everything leads up to that point. So when we write a conclusion, we have 3 paragraphs for this class. The first one is results with evidence. So this is one where you tell us what you did, and you tell me what it means. It's no good if you say, hey, I caught all these bugs and I put them in a jar and 5 of them are girls. So, I want to know, what does that mean? Errors. What are some things, right? Not everyone does everything perfect the first time. What are some things that you could fix and do it better next time? Science is cool in that you get second chances a lot of times. And if you don't fix it, maybe someone else will fix it. So if you find an error, maybe the students in next year's class won't have that error. Lastly, applications. This is famous for being the so what. Who cares? A lot of times you're in a class and you're doing a lab and you're thinking, why does this matter? This is your point to find out application. Is there money involved? Is there politics involved? Why is this important? For example, cancer is all about medicine. If you're doing research, you want to save lives. In this class, we're doing things, maybe it is practice. It's a practice lab to prepare you for the real world. But try to think of something that's a real world application. Why would somebody from HMMI, a huge research facility, Howard Hughes Medical Institute, come talk to you for your research in my class? So, here's one last question. In most stable water environments, population of Daphnia. So if it's in italics, that usually means it's a species. It's a type of animal. They're almost entirely female and reproduce asexually. However, males are observed in low oxygen environments or when food is scarce. Based on these observations, a researcher asserts that male Daphnia develop in response to unfavorable environmental conditions. Is this a result, a theory, a procedure, or a hypothesis? Take a moment. This one would be a hypothesis. It's not set up in an if-then statement, but you could make it one. If there is unfavorable environment, then male Daphnia, then males show up. That's your if-then statement. If there's an unfavorable environment, then there will be more males. So to wrap up, make sure you know the different steps in the scientific process. Even though I've showed it here as linear, most of the time it actually is a huge mess. And a great place to see that is the website Understanding Science from UC Berkeley. This shows you a couple examples of actual research from people that show you that science is not some perfect process. A lot of times you come up with a hypothesis and you test it out and it makes no sense. So you've got to go back and fix it. Another example would be you get to your conclusion and it's garbage. You can't write a PhD paper on that. You can't go to a professor and be like, this is what I found. Got to go back to the field. So remember, science is messy. Number 2, what is a hypothesis and how do you make one? Think about that and what the structure is. 3, compare an independent and dependent variable. Think about where you would put those on a graph. And lastly, distinguish between a hypothesis and a theory. This one's especially key, especially when talking about science in the public eye. So that's the scientific process. Lastly, I just wanted to touch on the 4 themes. So in this class, we're going to be talking about 4 themes, and that's how the year is going to be divided up. These are basically the big ideas for the rest of the year. And they break down to this. The first thing we're going to do after we finish introing bio in our first quarter is we'll talk about molecules to organisms. So molecules to organisms is the idea that you're focusing on something from small to big. So how do small things make up big things? This is kind of the idea behind Legos. How do you get from one Lego to a huge thing? We'll also talk about ecological interactions. So especially emphasizing on the idea of climate change and what we're doing to the environments around us. In heredity and variation, we'll talk about why you look like your parents. And we'll also hit on the major idea of mutations. Are they good? Are they bad? What do they do? And lastly, we'll hit on the biggest idea of all science, evolution and all the way of life. Remember I told you biology meant the study of life? So at the end of this year, we'll talk about evolution and how it impacts everything, from anything from medicine to health to most major decisions that we make that affect public policy. We'll also talk about the diversity of life, which includes things like plants and animals, and money pending. Hopefully get to do some dissections, but we'll see what happens. So hopefully you had a good time joining me as we took this little trip on the Intro to Bio. Hope you have a great day, and take care. See you next time.